The first presentation I listened to was a digital guide to the bees of northeastern Trinidad from Zoe Bedell, an undergraduate student. The main question in this study was how biodiverse are the bees in this area? This was important to figure out considering bees are a keystone species and biodiversity correlates with the health of an environment. This research was conducted in Toko, where the star is. The results of this study were found through setting up 12 sampling locations with several pan traps, a malaise trap, aspirators, and various sweep netting excursions. The results found a total of 148 specimens, which represented 22 different species. This presentation surprised me when hearing about the number of specimens and species found through this study. It's very interesting to learn just how biodiverse an organism as small as a bee can be. A connection I made between the presentation and our Fundamentals of Ecology class was the mentioning of bees being a keystone species, meaning that even though they may be small in population size, if they were removed from their environment, everything would be completely off balance and different. The next presentation I listened to was the impact of persistent entomopathogenic nematodes for biocontrol of corn rootworms on non-target arthropods in Texas, from Pragya Gupta, a graduate student. Corn rootworms, which is what these two are, this is when they're larvae and this is when they're fully grown, have a severe economic impact. Since they seem to have a resistance to most control methods, a new method being looked at is using their natural enemy to essentially kill them off. This enemy is known as entomopathogenic nematodes, or EPNs. The hypotheses for this study were that the EPNs would not only kill off their main target, the corn rootworms, but also non-targeted pests as well as beneficial insects. The small organisms in this picture are what the nematodes are, and essentially their main purpose in life is for killing insects. And so that's why this was a big worry, with them killing the beneficial insects. The results showed that EPNs had no adverse effects on beneficial insects, but they did cause a higher mortality rate in non-target pests. As an agricultural science major, I found this presentation to be one of the more interesting ones to me. I didn't know about corn rootworms and the big impact they have, so it was good to learn about new methods that help control them and also have no negative effects on beneficial insects. The last presentation I listened to was Exploring Plant Insect Networks Associated with Native Grasses in North Texas by Isabella Sibeldi, a graduate student. There has been a decline in pollinator insects on a global scale, and in order to improve these numbers, there's been an effort to improve pollinator-plant relationships on a local scale. Grasses may be one of the key plants in improving these numbers, and also with the help of arbuscular mycorrhizal fungi, also known as AMF, which is what's underneath this microscope. The experiment assessed whether AMF had any effect on pollinator insect networks with grasses in North Texas during the flowering period and the grass pollen productivity during the late flowering period. The process of running this experiment included randomizing plots under 4 conditions, including mycorrhizal inocula, nutrient load, and 5 species of native grasses. Soil, pollen, flower visitors, floral characteristics, pollinator visitation, richness, and abundance were collected. Amino acids, crude proteins, non-structural carbon, proximate and fiber composition, fatty acids, and micronutrients were harvested. The results were that the AMF only really affected pests and had little to no effect on beneficial insects. What intrigued me with this research was the connection I made to it from my soil and crop sciences class. In this class, it's been mentioned that plants and soils are often made healthy with the organisms that live in and on them, which was one of the main points of this report.